Hello, everyone. Let's dive deep into the molecular mechanisms, epigenetics, and experimental models that define fungal cell biology research today. That is actually the postdoctoral level research frontier for research oriented mechanistic and systems level insights. Cell wall remodeling and host adaptations, dynamic interface with the host. This is the real-time modulation via endosomal recycling, real-time remodeling studies using atomic force microscopy. This is highly plastic and immunologically active. Candida albicans masks the beta-glucan using Manan shields and ESCRT machinery. What is ESCRT? That is the endosomal sorting complexes. This machinery controls immune evasion. So composed of inner chitin beta 1316 glucans and outer manoproteins, GP1 anchored proteins. Cryptococcus and Candida dynamically alter glycosylation to evade PRRs. Chitin exposure controlled via endosomal sorting complexes. One thing you have to remember in fungi, cell wall is the outermost covering. Inside that is the cell membrane, and then cytoplasm is covered by the cell membrane and then covered by the cell wall. But in human, there is no cell wall. And in plants also, outside the outermost covering is the cell wall and then cell membrane. Inside is the cytoplasm. Cryptococcus and Candida dynamically alter glycosylation to evade PRRs and chitin exposure controlled via endosomal sorting complexes. Extremely sorry for the noises, the disturbances. Now let's move on to the next slide. Some recent interesting advances are there. Candida albicans uses beta-glucan masking via endocytic recycling and Manan shields. ESCRT pathway modulates cell wall architecture under stress and host immune evasion by dynamic remodeling of PAMs. So if those gets remodeled, then PRRs, which were ready to neutralize the PAMs, they were no more ready for neutralize the remodeled PAMs. So thus they escape the host immune system. And future direction, wall proteomics plus glycomics to develop diagnostics and vaccines. Polarized growth and Spitzenkorper, that is the vesicle trafficking. Spitzenkorper, those are the vesicle organizing center present at the hyphal tip, coordinating exocytosis. Actin and microtubule mediated transport of chitin synthase, glucan synthase, lipid rafts and polarity complexes, septin ring, exocyst complex forming proteins drive apical extension. Those you have to remember for the postdoctoral researchers. Key proteins are TEA1, FOR3, BNIL, exocyst components like SEC3 and EXO70. SPK, that is a Spitzenkorper in short. SPK dynamics regulated by TOR and Rho GTPase. Example, CDC42 and RAC1. Those are vesicle coordination center at the hyphal tip I have already mentioned. Again, sorry for the disturbance. Now coming to the organelle interaction and autophagy. This is also known as organelle crosstalk. That is the mitochondria peroxisome crosstalk. This is interaction for virulence. Now, inter-organelle communication, mitochondria peroxisome and endoplasmic reticulum Golgi apparatus. Mitochondria peroxisome crosstalk that is required for beta oxidation and ROS handling in virulent fungi. Ustilagomedis and Candida tropicalis rely on these axes during infection. Vacuole mitochondria communication linked to aging and antifungal stress response. Autophagy essential in pathogenesis, ATG8, ATG40 in Magnapathy, and live cell imaging shows vacuolar fragmentation in stress. So imaging tools for study these organelle crosstalk. live cell imaging, biosensors. What are the biosensors? GFP tagged biosensors, threat based pH indicators, and live cell confocal microscopy. ROS means reactive oxygen species. GFP tagged biosensors, where GFP is the green fluorescent protein and FRET is the for fluorescence resonance energy transfer or for Forster resonance energy transfer. Now, next is the fungal cell cycle and nuclear dynamics. Closed mitosis occurs in fungi. A nuclear envelope does not break. Asynchronous nuclear division in multinucleate hyphae and chromatin accessibility modulated by SWI or SNF and RSC complexes. And centromere studies in Candida albicans and Aspergillus show plasticity and neocentromere formation. Now, a few lines for what is SWI SNF. This is the switch sucrose non-fermentable. That is, in pathogenic fungi, the SWI SNF complexes involved in regulating processes like hyphal differentiation, which is very much important in virulence, and responses to environmental stresses, which are important for the fungus to cause fungal diseases. Okay. Now, fungal signal transduction. TOR and PK pathways link nutrient sensing to morphogenesis. And MAPK cascade control cell wall integrity, osmotic stress, and calcium calcineurin CRZ1 axis causing the antifungal resistance. So few words in short for TOR pathway. What is TOR pathway? The full form is the target of rapamycin pathway. It is highly conserved signaling pathway that acts as a central regulator coordinating essential cellular processes like ribosome biogenesis, protein synthesis, amino acid transport, endocytosis, autophagy in response to environmental stresses. And what is PK? PK is the protein kinase A pathway, which is a critical cell signaling pathway that starts with an extracellular outside the cell signal like a neurotransmitter binding to the GPCR, protein coupled receptor. Now, what is MAPK? MAPK cascade control. MAPK is the conserved signaling pathway which control essential processes like growth, stress response, pathogenicity by relaying extracellular signals through a three-tiered molecule. What are those? MAP, triple K, MAP2K, and MAPK. To ultimately, they regulate gene expression via transcription factors. MPK1 means mitogen activated protein kinase 1. This is the MPK1. And HOG1, this is also a key mitogen activated protein kinase 1, which plays crucial role in stress response, stress resistance development, and pathogenicity. And what is calcium calcineurin CRZ1 axis, which is a conserved signaling pathway specifically for fungi, where an increase in intracellular calcium inside the cell when calcium concentration increases, it activates the phosphatase calcineurin, which then dephosphorylates the transcription factor CRZ1, and then antifungal resistance, etc. Different activities take place. The fungal autophagy, stress and aging. So autophagy is vital for what? What are the purposes for autophagy? For sporulation, nutrient starvation, host survival. Autophagy genes are ATG8, 11 and 40. Mitophagy and pexophagy protect against ROS, reactive oxygen species, NAD, host killing. reactive oxygen species, sorry, this is typing error, and host killing. Genetic screening via CRISPR libraries in Candida albicans, Aspergillus nidoulans can be done. Now, phase separation and epigenetics. Phase separation regulates transcriptional condensates in Candida. Chromatin modifiers like SET1, HTA1 alter resistance and virulence genes. Centromere plasticity affects genome evolution and karyotyping switching, liquid-liquid phase separation. Discovery for transcriptional hubs and stress granules form via LPS in Candida glabrata and Neurospora crassa. Involved in mRNA storage, epigenetic silencing. What are the experimental tools? FRAP, optogenetic LLPS, perturbation. Regulates the what are the purpose of phase separation and epigenetics? Regulates stress responses and antifungal tolerance. That is the most important. Now, extracellular vesicles or EV, they carry RNA, virulence factor, lipids, use in immune modulation, drug resistance, and promising diagnostic biomarkers. For extracellular vesicles, ongoing researches are there for Cryptococcus. Epigenetics and uh, transcription plasticity. Histone modification regulate pathogenic transitions. East hypher switch, white opaque switching in Candida albicans, chromatin modifiers like SET1, HD1, epigenetic memory of drug exposure, fluconazole persistence via transient H3K27AC. Those are the mechanisms inside the fungi for nothing but for growth evasion and most importantly for antifungal resistance. Now, what are the latest tool in fungal cell biology? CRISPR-Cas9, base editing and prime editing in Candida aspergillus, super resolution microscopy for SPK and septum visualization, and fungal single cell RNA sequence for heterogeneity studies. Host fungus interface models, organoids infected with fungi to model pathogenesis, live zebrafish and murine imaging for EV trafficking, and immunometabolism studies show lactate and hypoxia modulate fungal virulence. Molecular antifungal resistance mechanisms. We know the drugs, their targets on cell wall and cell membrane, but at the molecular level, a postdoctoral researcher must know. Azoles, ERG11 point mutation, upregulation of efflux pumps like CDR1, CDR2, and MDR1. Aneuploidy and isochromosome formation, especially in Candida auris. For echinocandins, FKS1 and 2 mutations, those are the hotspot 1 and 2. For photodicin B, reduced ergosterol via ERG2-3 loss. Emerging resistance tracking tools are genomic surveillance, metagenomic signatures, and resistance gene panels. Now, tool and application. A chart in chart form. Super resolution microscopy, that is vesicle dynamics, septal pore structure, CRISPR-Cas9 base editing, prime editing, to conduct the gene knock in or out in Candida mucorales trichoviton, single cell RNA sequencing, heterogeneous stress response uh, study, organoids and microfluidic chips to study in vitro host fungus interaction, and zebrafish larval models, live imaging of fungal dissemination and immune evasion. This is very interesting one. Now, systems biology and fungalomics, transcriptomics and proteomics under antifungal stress, dual RNA sequence to study host fungus interaction at cellular level, metabolomics identifies nutrient acquisition and metabolic reprogramming in infection, integrated network analysis using cytoscape, StringDB, and K-base path or pathway tools. SpringDB is nothing but a search tool for the retrieval of interacting proteins or genes. Now, possible research avenues in 2025 host fungal vesicle trafficking crosstalk in macrophages, LLPS in nuclear stress bodies during antifungal exposure, fungal EVs as diagnostic biomarkers in bloodstream infections. This is very important. And CRISPR-I screens to discover essential genes in non-model fungi and phase-specific expression maps of dimorphic fungi during morphogenesis. So few related topics for MD final exams and Viva. One important is comparison of azole resistance mechanisms, Candida albicans versus Candida auris. Efflux pumps, target site mutation, those are different. Upregulation of ERG genes, also different. Biofilm formation and clade variability, those are different. So you have to study and remember this. But this chart is very helpful. Fungal dimorphism. From this, we will discuss from the next session. So, learn up to this. Hope you have enjoyed the session. Thank you. Please like, share, and subscribe my channel. Have a good day. We'll meet again very soon.